The reptilian gene is a fascinating and complex subject, representing millions of years of evolutionary adaptation. Its distinct characteristics have enabled reptiles to thrive in diverse and challenging environments, from arid deserts to lush rainforests. The study of the reptilian gene provides insights into the fundamental mechanisms that govern not only reptilian physiology but also the broader spectrum of vertebrate genetics. This article delves into the unique properties of the reptilian gene, its evolutionary significance, and its implications for understanding biodiversity. Reptiles, as one of the oldest groups of vertebrates, have undergone a series of genetic transformations that have allowed them to adapt, evolve, and persist across geological epochs. Their genomes contain clues about survival strategies, behavioral traits, and physiological responses that are unparalleled among other vertebrates. Understanding these genes helps researchers trace the origins of various genetic pathways, shedding light on the evolutionary history of reptiles and their divergence from other groups such as mammals and birds. The unique anatomical and physiological features of reptiles, such as their scaly skin, cold-blooded metabolism, and regenerative abilities, are deeply rooted in their genetic makeup. Unlike mammals, which are endothermic, warm-blooded, and possess fur, reptiles are ectothermic, relying on environmental heat sources to regulate their body temperature. This key physiological difference is governed by a distinct set of genetic expressions that define their metabolic pathways. One of the primary genes responsible for this trait is the LHX9 gene, which influences thermoregulation and energy conservation. In reptiles, this gene plays a crucial role in optimizing energy use in varying climates, allowing them to survive in both extremely hot and cold environments. The scaly skin, which is a hallmark of reptilian biology, is regulated by keratin genes unique to this group. These genes, such as KRT9 and KRT14, control the production of specialized proteins that form the hard, protective scales, providing a barrier against dehydration and physical injuries. Additionally, reptilian genomes show a high degree of genetic plasticity, particularly in relation to their regenerative capabilities. Certain species, like the gecko and the green anole, possess genes such as WNT and TGF-beta pathways that facilitate tissue regeneration. These genes allow them to regrow lost tails and limbs, a feat rarely seen in mammals. The behavior and cognitive abilities of reptiles, while often considered less complex than those of mammals, are nonetheless influenced by their genetic frameworks. Reptiles display a range of behaviors from territoriality and mating rituals to navigation and problem-solving skills, all of which are dictated by neurological gene expressions. Key genes such as BDNF, brain-derived neurotrophic factor, and NRG1, neuregulin-1 are known to regulate neurodevelopmental processes, influencing learning, memory, and social behavior in reptiles. For instance, research has shown that some reptiles, like certain lizard species, are capable of spatial learning and have the ability to navigate mazes. This contradicts the stereotype of reptiles as solely instinct-driven creatures and suggests that their cognitive faculties are more advanced than previously thought. Furthermore, the expression of genes such as DRD4, a dopamine receptor gene, plays a role in modulating reptilian temperament and responses to environmental stressors. Variations in this gene can lead to differences in behavior between aggressive and docile individuals, shaping the social dynamics within reptilian populations. The reptilian gene has undergone significant evolution, marked by both conservative and divergent changes that have allowed reptiles to occupy a variety of ecological niches. Molecular analysis reveals that certain genes, such as the HOX gene cluster, have remained remarkably stable over millions of years. The HOX genes are responsible for body segmentation and limb development, ensuring that reptiles maintain their characteristic body plans despite changes in size and environment. However, other genetic pathways have diverged considerably, giving rise to unique adaptations in different reptilian lineages. For example, the sonic hedgehog SHH gene, which is critical for limb and digit formation, shows unique mutations in snakes, resulting in the loss of limbs, a defining trait of this group. In contrast, chameleons have evolved specialized visual genes like otoferlin, enabling them to process color information with exceptional precision, which is crucial for their predatory lifestyle. The divergence between reptilian and mammalian genes is particularly evident in the TBX5 and PITX1 genes, which influence limb morphology. In reptiles, these genes contribute to the development of elongated bodies and reduced limbs, a feature seen in species like skinks and snakes. This genetic divergence has allowed reptiles to explore niches inaccessible to other vertebrates, from burrowing underground to gliding between trees. At the molecular level, the reptilian genome is characterized by a rich array of gene families that control various physiological processes. One of the most prominent is the cytochrome P450, CYP family, which is involved in detoxification and metabolic regulation. Reptiles possess a greater diversity of CYP genes compared to mammals, allowing them to process a wider range of dietary compounds, including toxic plant and animal matter. The CYP gene family's expansion is thought to be an adaptive response to the varied diets of reptiles, ranging from herbivory in tortoises to the carnivorous habits of snakes. This genetic versatility is a key factor in the ecological success of reptiles, enabling them to exploit diverse food sources without succumbing to toxins. Another crucial aspect of reptilian molecular biology is the role of microRNA, miRNA, in regulating gene expression. miRNA are small non-coding RNA molecules that bind to messenger RNA, mRNA, and control the translation of genetic information into proteins. In reptiles, miRNA are known to modulate various processes such as development, metabolism, and immune responses. For instance, specific miRNA, like MIR1 and MIR206, involved in muscle development and repair, contributing to the extraordinary physical abilities of certain reptiles, such as the powerful tail whips of iguanas or the rapid limb movements of lizards. Additionally, miRNA play a role in temperature-dependent sex determination, a unique reproductive strategy seen in some reptiles like turtles and crocodiles. The expression levels of certain miRNA change based on the temperature of the egg incubation environment, influencing whether the embryo develops as male or female. This complex interplay of miRNA and gene regulation highlights the sophisticated genetic toolkit that reptiles possess, allowing them to fine-tune their physiology and adapt to changing environments with remarkable precision. The genetic resilience of reptiles is one of their most intriguing features. Their genomes are equipped with adaptations that provide resistance to diseases and environmental stressors. One of the primary genetic factors contributing to this resilience is the presence of unique immune system genes, such as toll-like receptors, TLR, and major histocompatible complex, MHC genes. These genes are essential for pathogen recognition and immune response, enabling reptiles to fend off a broad spectrum of infections. Moreover, the P53 gene, known as the guardian of the genome, is highly conserved and active in reptiles. This gene is responsible for regulating cell cycle checkpoints, DNA repair, and apoptosis, programmed cell death. In reptiles, the P53 pathway is more robust compared to mammals, providing enhanced protection against cancer and other age-related diseases. This genetic advantage is one reason why some reptilian species, such as tortoises, have exceptionally long lifespans and relatively low incidences of cancer. Additionally, reptiles possess genes that confer adaptability to extreme environmental conditions. For example, desert-dwelling reptiles, like the Gila monster, express unique variants of the aquaporin genes that allow them to efficiently manage water retention and minimize dehydration. Similarly, reptiles that inhabit cold environments, such as the painted turtle, have evolved specialized proteins known as antifreeze glycoproteins AFGP, which prevent ice formation within their cells during hibernation. These genetic adaptations not only ensure survival but also allow reptiles to thrive in a wide range of habitats, from the arid deserts of North Africa to the frozen tundras of the Arctic. Reptiles are among the most diverse groups of vertebrates, with an evolutionary history that dates back over 300 million years. The divergence of reptilian genes from those of other vertebrates, such as mammals and birds, can be traced through comparative genomics. By analyzing the differences and similarities in gene sequences, researchers have identified several key genetic pathways that set reptiles apart. One significant divergence is seen in the SOX gene family, which plays a pivotal role in sex determination and gonadal development. While mammals rely on the presence of the SRY gene on the Y chromosome to determine maleness, reptiles exhibit a broader array of genetic mechanisms. In some species, like the American alligator, temperature-dependent sex determination is influenced by SOX9 expression. The lack of a strict chromosomal sex determination system in these reptiles suggests that they have evolved alternative genetic pathways for reproductive success. Another area of divergence is in the FOXP2 gene, often referred to as the language gene. In mammals and birds, mutations in FOXP2 are linked to speech and vocal learning. In reptiles, however, this gene shows a unique expression pattern associated with territorial and courtship behaviors, suggesting that it has been repurposed for social communication rather than vocalization. This repurposing highlights the plasticity of the reptilian genome and its capacity to evolve novel functions from existing genetic frameworks. Reptiles are often seen as solitary creatures, but their social behaviors are more complex than they appear. Genetic studies have revealed that certain genes, such as arginine vasopressin receptor 1A, are linked to social bonding and aggression in reptiles. Variants of this gene are associated with different levels of territoriality, mating strategies, and parental care. For example, in the green iguana, individuals with higher AVPR1A expression tend to be more territorial and engage in more aggressive displays, while those with lower expression are more tolerant of other individuals. This genetic basis for social behavior suggests that, like mammals, reptilian social structures are influenced by underlying genetic mechanisms that shape their interactions and hierarchies. The oxytocin and vasopressin receptor genes, which are well studied in mammals for their role in bonding and social behaviors, also exist in reptiles, but with different expression patterns. In reptiles, these genes are more closely tied to reproductive behaviors and mate guarding rather than long-term pair bonding. This distinction provides insights into how reptiles have evolved social strategies that differ from those of mammals, emphasizing the diversity of genetic influences on behavior. Despite the wealth of knowledge gained from reptilian genetic research, several controversies remain. One of the most debated topics is the potential for genetic manipulation of reptilian genes for conservation or biomedical purposes. As reptiles face increasing threats from habitat destruction and climate change, scientists are exploring the possibility of using gene editing technologies like CRISPR to enhance their resilience. However, ethical concerns arise regarding the manipulation of wild populations. Altering genes to increase disease resistance or change reproductive strategies could have unforeseen ecological consequences, potentially disrupting natural evolutionary processes. Critics argue that such interventions could lead to a loss of genetic diversity and the homogenization of reptilian populations. Another area of contention is the classification of certain genetic mutations in reptiles. For instance, the presence of pseudogenes, genes that have lost their original function, in the genomes of some snake species has sparked debate about the role of these genetic relics. While some researchers view pseudogenes as evolutionary dead ends, others believe they may still play a role in regulating gene expression and could be reactivated under certain environmental conditions. These controversies highlight the complexities of reptilian genetics and underscore the need for further research to unravel the full implications of these genetic phenomena. The future of reptilian gene research holds great promise for both scientific understanding and practical applications. Advances in genomic sequencing and bioinformatics are enabling researchers to map the entire genomes of various reptilian species, providing a comprehensive view of their genetic architecture. Such studies are revealing previously unknown gene families and regulatory elements, offering new insights into the evolution and adaptation of these ancient creatures. One promising avenue of research is the exploration of epigenetic modifications in reptiles. Epigenetics, which involves changes in gene expression without altering the underlying DNA sequence, is known to play a role in temperature-dependent sex determination and developmental plasticity in reptiles. Understanding these epigenetic mechanisms could lead to breakthroughs in reptilian conservation, allowing scientists to predict how populations will respond to environmental changes. Additionally, the study of reptilian genes may have applications beyond herpetology. For example, the unique regenerative abilities of some reptiles could inspire new approaches to human medicine, particularly in the fields of tissue engineering and regenerative therapies. By identifying the key genetic factors that enable reptiles to regenerate limbs and spinal cord tissues, researchers hope to develop treatments for spinal injuries and other debilitating conditions. The reptilian gene is a testament to the power of evolution and the adaptability of life. From their unique physiological traits to their complex behaviors, reptiles showcase a diverse array of genetic adaptations that have allowed them to thrive for millions of years. Understanding these genes not only enriches our knowledge of reptilian biology but also provides valuable insights into the broader processes of vertebrate evolution and adaptation. As research continues to uncover the intricacies of the reptilian genome, the potential for new discoveries remains vast. Whether through the study of genetic resilience, behavior, or disease resistance, the exploration of reptilian genes promises to deepen our appreciation for these remarkable creatures and contribute to the ongoing quest to decipher the genetic code of life itself.